First, let me start by sending the link to the slides. So if you want to follow along and copy paste stuff out of them, I'm going to put it in the Zoom chat right now. Sorry, that's the wrong part of the line. Okay. So the talk today is improving ontology interoperability with biomappings. So here's the link for the slides. And then if you want to follow along in the code, this is also the link for that. So the goal for today, there's 4 parts. The first 2 are motivation. I want to show you how I sort of ended up thinking about mappings and trying to build software to help me create more of them, because it turns out a lot of mappings between various resources are incomplete. And then also give you a little bit more motivation from the high level, because we're not just doing mappings for mapping's sake, but it's typically part of some other kinds of workflows, like building knowledge graphs, which also are part of bigger workflows for other translational medicine applications. Along with that, there's a dichotomy between the first-party mappings that come with resources from the people who actually maintain the semantic space that those mappings are incident to. And then there's the third-party or second-party sort of version of it, where we might be able to create our own and add them back. And so this has sort of been a theme for a lot of the talks that I've given in the last year or so, sort of how does the community participate in the curation and contribution of mappings to support mappings that already exist. So for example, in Monarch world, there's Monarch mappings to mesh, which aren't complete. And I've curated some myself and contributed them back using this biomappings workflow I'm going to talk about today. And so then the third and the fourth part is to teach you about the biomappings workflow, both the philosophy and actually the technical side, so you can run it yourself and actually do some curation. It turns out that curating is really fast when you have a curation interface to help you do it. And if you've already gotten curations sort of prepared for you based on some kind of semi-automatic workflow, you spend a lot less time doing this kind of curation. And so I'm actually going to also come back, if this is interesting, maybe I'll poll the group if you actually want to see a demo on how you can do your own pipeline to generate lexical predictions. So maybe if there's someone in the audience who has an interest in mapping 2 different resources, we can go through how you would actually generate those predictions yourself, and then they get kind of put into the biomappings workflow. Okay, so my career, I think, has been sort of a descent into the lowest level of hell. I keep telling people that I ask them, do you know Dante's Inferno? And that's sort of this allegory as you go further and further into hell, things get worse and worse and worse. And way before I got into this inferno, I was a synthetic organic medicinal chemist, thinking I was going to make the next medicine in the lab. And then I became a computational scientist, and I started doing networks medicine. And so I was doing machine learning on graphs with PyKeen. And then I started realizing we need to make better networks. So I got into stuff like natural language processing and data integration like Indra. And to support doing that, we actually have to have good mapping. So that's what this talk is about. All the way at rock bottom in the Dante's Inferno allegory of bioinformatics, I ended up thinking about stuff about the bioregistry. So if you didn't hear my talk last week, you can maybe go back to that and think a little bit about identifiers and the deepest, darkest secrets of bioinformatics. But anyway, let's take a big step back and let's talk about 3 parts of talking about information extraction. So given some mechanistic biology, like PTPN11 dephosphorylates PTK2, maybe you see this in a paper somewhere. If you want a machine to be able to do something with this, usually the first aspect of information extraction, something like named entity recognition. So you probably want to identify that PTPN11 is this gene somewhere. So you have a named entity recognition software that does that. The second part is you want to take that name and ground it to some vocabulary, like this is a protein, so we're going to ground it to the UniProt vocabulary. And then the last part would be named entity standardization, where if you have lots of different pieces of information coming in where you've done grounding and named entity recognition, you probably want to make sure they're grounded to a standardized vocabulary. So let's map this from UniProt to HGNC. And so I'm using this kind of colloquially, and I'm going to define it a little bit more specifically, but we need to have high quality mappings from one semantic space like UniProt to another one like HGNC if we want to do this named entity standardization. And while we're doing this, we're going to have to think about the kinds of mappings there are, whether they're broad, narrow, and if they're complete or not. So what is a semantic mapping? First, I'm going to refer you back to some of the really nice SESM talks that actually define this in a more formal way. But most of the time when we're talking about semantic mappings, we're saying 2 things are equivalent to each other. So for example, ethanol appears in the KEBI vocabulary. It also appears in drug bank. Both of them assign a different identifier to this same like absolute concept. But we want to know that if you're in the KEBI world, it has this identifier 16236. And if you're in the drug bank world, it has this other identifier. And both KEBI and drug bank maintain mappings from their semantic space to other semantic spaces. So KEBI has drug bank mappings, it has Metaseq mappings, HMDB mappings, and so on. And then drug bank also has mappings to those and other resources. So like the ones that they map to might be different. So yeah, like I mentioned, there's lots of different types of ways of describing mapping, so different namespaces or different vocabularies for describing the kinds of mappings, but most of them boil down to being stuff like exact mappings, broader mappings, narrower mappings, close or related mappings. I'm going to just talk about SCOS, like the SCOS exact match and narrow and close and broad matches. I'm going to just refer to those today. But in the OWL world, there's also this idea of like equivalence. Those have their own little nitpicky bits to them, but for typically the applications that I'm doing, like they can be kind of considered equivalent. Yeah, so by the way, anyone can feel free to just speak up and interrupt because it's a little bit tricky for me to check the chat while we're going. Okay, so like I mentioned, I just even maybe committed heresy already by saying like that I consider some of these different predicates to be the same, even though they really aren't. In my applications, I want to like combine data and knowledge. So let's say last year we were working with this cancer cell line encyclopedia data, which is data measurements, and we have knowledge that's encoded in these causal mechanistic statements in like a knowledge graph that we get from Indra. And well, they use different vocabularies to identify cell lines, and so it turns out there's actually a lot of vocabularies that identify cell lines, and the arrows on this diagram are sort of going from one vocabulary to another if that vocabulary, so CCLE, it maintains mappings to DepMap, and DepMap maintains mapping to Cellosaurus, and Cellosaurus maintains mappings to EFO, to Brenda, and to DepMap. So it turns out there's no CCLE to cell ontology mappings, and our data is from CCLE, so it has CCLE identifiers associated with it, and our knowledge is grounded into cell ontology terms. So like you have to kind of play this game where you go mapping to get to the equivalent entities so you can attach your data to knowledge. And so you can go directly or you can use inference, but either way your mappings are still incomplete most of the time. So that's one motivation, is I want to complete my mappings so I can combine my data and my knowledge or integrate different resources. A little bit more specific view on this thing, which shows actually the numbers between CCLE, DepMap, Cellosaurus, and EFO. And so, yeah, you can add inference to this, and that's still not doing the job. So I'm going to gloss over this slide, actually. So what does this look like in the knowledge assembly world? Well, let's say we have a database that uses KEBI terms as its subjects. And so let's say we've got our relationship from this KEBI term ethanol, and we've got some predicates which aren't really labeled well. Actually, sorry about this. I really don't like this picture. So we've got, let's say n different triples that have n different objects. And then we have another database that uses drug bank as its identifiers. And so it also has some other statements, and we want to combine those two together. So we need to make sure that all of the terms that either say KEBI or drug bank get mapped to the same one. So let's say that our priority prefix is KEBI. So anytime we can map a drug bank term to a KEBI term, we want to also update those triples. So that would look like this. So now we remap all of those triples to have the KEBI term as the subject instead of drug bank one. And why do we want to do this? We don't want multiple nodes of representing the same thing showing up in the same knowledge graph. Okay, so what are the big problems here with mappings? The first one I mentioned from this big hexagon of cell line resources is that first-party resources don't have complete mappings to other resources, or maybe they're completely gone. For example, there's no mappings between mesh terms and KEBI terms. And I'm sure everybody's loved to use mesh before for searching the literature. Some databases like the chemotoxicogenomic database even use mesh as its identifiers in its triples. And so either they're incomplete or they're completely missing. Predicted mappings typically don't get persisted. So people want to fill in these mappings, and some people use automated methods for doing this. but they don't save the results. And even worse, we don't really have curation tools that are great for predicted mappings. And so, yeah, I pick on the ontology alignment initiative, which I think Nico might be able to say this is improving now. But they've been using the same task of aligning the foundational model of anatomy ontology with SNOMED CT. Every year, they've had people publishing new prediction methods for mappings between these two resources. It's for 15 years. In my opinion, they should have made the predictions, curated them, and then sort of moved on because we don't need to keep building new methodology. We actually just want the content. So this isn't a talk about prediction methodology. This is only a talk about how to use the results that come out of it. And yeah, so ideally, once you do this prediction and curation, you actually reincorporate the results into the upstream resources. So if I make new curations of Mondo to mesh term mappings, like I shouldn't just hoard them. I should just contribute them back to Mondo, and Mondo has a nice permissive license. So like then everybody gets to benefit from this. And all this being said, we should do this sort of in this open code, open data, open infrastructure, O3 way. So we're going to start saying O3 from now on for this. All right, give a second for everyone to digest that, and I'll check the chat. Okay, Tiago has rightly said that I miss speaking about cell line ontologies. So I've mentioned cell lines and cells sort of interchangeably throughout this talk because a lot of the resources cover both of them. All right, so what I'm not going to cover today is actually SESAM itself. So like the standard for storing mappings. And actually, I'm not going to go into the main problems with like why semantic mappings are having insufficient metadata and like this problem. That's also the case for tools like ontologies like Mondo. I'm also not going to talk about this like large scale assembly of mappings that combines primary and secondary sources. This is the next step on top of doing biomappings is to be able to assemble all of these to like make your knowledge graphs. But I'm not going to go into that application while we're talking about this today. Today, we're just going to focus on the biomappings workflow. So this is like a philosophy. I'm going to describe the philosophy sort of I have informally already. And then after this, we're going to go right into like how can you as the listener like use this yourself, whether it's for curating more mappings for Mondo, because this is like the project that you're part of. Sorry, I keep mentioning Mondo as the example, but this could be like any ontology that you're working on. Okay, so the first thing you have to do is like get all of the content that you need. So if you want to make a comparison between Mondo and Mesh, you should be able to download all of the newest terms from both Mondo and Mesh. Then you should have some kind of tool that predicts the mappings between them. So here I'm going to talk about Gilda because it's a like amazingly fast and accurate lexical matching tool. I'm aware that Oak also has something like this, though I haven't used it myself. There's also other methods for predicting mappings that aren't lexical methods. So I'll mention very briefly later that PyKing can be used for knowledge graph-based alignment, or you can use whatever kind of custom scripts you want. After you do that, you should sort of filter down and pick a scope for your curation. So one of the important things to do is like don't recurate stuff that already exists. So if you use Mondo, it already has mappings to Mesh, so you should filter out all of the existing mappings before going to curate new ones because you don't want to waste your effort on stuff that already exists. You might also want to do filtering based on the cardinality. So maybe you don't want to spend time curating mappings where it's been predicted multiple possible mappings. Then you should go about curating them. And it's not just that you want to curate and get good, correct content, but it's actually worth keeping curations that are incorrect. It's worth keeping information about sort of non-trivial, incorrect, negative mappings. And then I have also found during this process that it's useful to be able to mark mappings as unsure and sort of leave them for later. And then the last thing, after you've done all your curation, you should use the wisdom that you've gained through that curation to potentially improve the prediction pipeline. So if you're using a canned lexical matching pipeline, this is a little bit more tricky, but if you have some kind of custom script, you may realize there are certain systematic issues with the way mappings are done that you can use to improve the way that the mappings are generated, because this actually never ends. Typically, resources are creating new entities, and you're going to want to do this cycle over and over and over again, though the burden may lessen over time. All right, any questions until now? Good. Nico says it all makes sense because he's heard me give the talk many times. Anybody else? If not, then I'm going to briefly talk about how the prediction for lexical matching works in case you're not familiar. This is the way that we as humans typically think about matching things, because we look at their names and their descriptions and maybe their synonyms, and we see if those are the same. So I guess I just buried the lead because that's what lexical matching is. All right, we're going to skip this. I already mentioned existing matchings. So the way Gilda works, it indexes both the labels and the synonyms from entities. It uses some really interesting but simple algorithms for creating variations of synonyms and labels, and then it indexes them with some kind of string normalization. And then, yeah, we do this filtering I mentioned. We perform this all by all comparison. So yeah, actually, there's not much to say about this. It's lexical matching is fast, it's simple, and it's interpretable. So when you have curations that are created this way, predictions that are curated this way, excuse me, then it's typically easy to go through and curate them. So let's look at a couple examples just to illustrate. So here we've got a Kebi term for 3,4-dihydroxybenzoate. So here's the molecule up on the top right. And it actually is the same in mesh. It has exactly the same label. So that's a really easy one. You can easily curate that. When we have similar labels, so what's going on here? We've got actually a difference in capitalization, but basically it's the same thing. So you can see that's highlighted. This is also very highly easy to score. You can have concepts with different labels, but we have synonyms that match. Okay, and this is, yeah, all of these different kinds of matches are scored by the Gilda algorithm. So then you get a number between 0 and 1, with 1 being like they're an exact text match and everything else in the middle. I already mentioned that Pykean and sort of graph machine learning can be used to do this entity alignment, but I'm going to kind of skip through that. If anybody's interested in this, we can come back to it at the end. All right, so curating mappings.
Charlie, there's a question from Nico for slide 24. He says, in this case, slide 24, are they the same?
Yes, all the examples I showed were the same.
Okay.
So if you have some chemistry domain knowledge, this is actually a pretty this is like an IUPAC kind of name, and this is more of a classic. The Kebi name is a IUPAC style name, and then the mesh name is more of a colloquial name. So these are indeed the same molecule.
Okay, thanks.
Okay, so I'm gonna have a question for the audience now. So I can go directly into the demo, or I can kind of give the end results of what's in biomappings before giving the demo. So before you answer that, I'll just show you. Here's kind of what the curation interface looks like. The demo today, and let me check the time. I think I'm going to spend 20-25 minutes on the demo, and it'll be completely interactive. When we do the demo, people can just get on the line and ask questions. I'm going to show how to get this thing up and running and how to use it, and then how to do the curations and then make contributions. After that, so this is what we're going to follow for the demo. After that, I've got, I think, maybe 5 or 6 slides just with sort of a wrap up. So are we ready to do the demo now, or do you want to hear the wrap up and then we could start the demo and completely go until the end of the hour?
I would suggest that, yeah. So leave enough time for doing the wrap up first. So I would say wrap up first and then do the demo afterwards. And then we can take as much time as we have. Okay,
so all right, let's imagine we're going to get back to the demo where we actually do all these things, and then the wrap up. So biomappings is a lot of things. It's a workflow, it's some code, it's itself a database. So inside biomappings, there's already mappings that have been curated. So there's over 10,000 manually curated correct mappings. There's over 1,500 verified incorrect mappings, so non-trivial incorrect mappings. And then we've got 40,000, more than 40,000 predictions sitting around waiting to be curated. And this comes from 60 different vocabularies, both across sort of molecular biology, chemistry, pathology. There's even some stuff that's kind of way out there. So various parts of the obo foundry that cover experimental techniques, agriculture. You can try to look down the list and see what you can see. Yeah, and so the point of all this stuff in my research is to improve the constructions of knowledge graphs. But there's lots of reasons why you would want to build up consistent sets of entities when you want to combine different data together for other reasons besides just building knowledge graphs. And then again, I want to point out this, keeping these negative mappings, these non-trivial ones, are really precious. We can be using them for machine learning. And these are techniques that people haven't touched before, because I don't think there's any databases of negative mappings. So I don't know, maybe biomappings is the first resource that actually took advantage of this part of the SESM standard to create these SESM files that have the not modifier in the predicate modifier column. And the other thing is the negative mappings help us reduce curation burden. So every time we generate a new set of predictions, we can already say, hey, look, these have been curated as negative. Like, don't make people curate them again. Whereas if you don't keep track of negative mappings, then people keep wasting their time on this. By the way, these are called zombie mappings, the ones that keep coming back. So we want to reduce the zombie mappings. All right. In addition to all of this,
Charlie,
yeah.
There's a question. Can you share what is species specific?
Oh yeah, that's an interesting one. So like I think I was using these for KEGG. KEGG has pathways that are not species specific. And then Wiki Pathways has pathways that are species specific. They represent the same pathway, but one that is not specific for a species and the one that is. Whereas the relationship between two of the same, say two representations of the same pathway in different species would have a different predicate, like an orthology predicate. Yeah, so we can look at examples of those later if that wasn't clear.
No, it's clear. I just wanted to point out that we have also now a vocabulary for cross-species mapping specifically. So
yeah, I was involved in that conversation, and
Yeah.
there's a predicate for this thing that I was just talking about. I got shot down. I wanted to propose one of those. So
okay.
For now, this is yeah, not a very well semantically defined predicate, but at least yeah, there's an explanation within biomappings for it. Okay. So given that we've got all this stuff, we've got this O3 thing going on with open code, open data, open infrastructure. We have the benefit that we can use all this continuous integration infrastructure for doing, quality assurance testing, for generating summaries, for generating quality control analyses. So, it's really useful to generate charts like this. So this one just shows like, what's the size of all the components in the mapping network that gets induced by all the biomappings content. So if you see like you've got a component that has 10 edges in it, you're like, oh, that's interesting. Maybe we've got some duplicates going on in there, or a number of duplicate prefixes. This is actually a little bit more specific. So there's a couple components that contain multiple entities from the same semantic space. So usually every semantic space only has one term or one identifier for the same concept. So if you find that there's duplicate prefixes within a click or component of this mapping network, then maybe some things are mapped to each other that shouldn't be. Maybe you've got some many to one or one to many or many to many mappings going on. And then you want to like review those and then use a different predicate besides exact matches in your mappings. And so like, yeah, you can go back. Typically the curation in biomappings is finding exact matches, but you can also curate it as different kinds of matches that are broad or narrow. And then one example of this where we found Ulster pollones and Ulster pollone, like this class of chemicals, and then like the specific prototypical instance of the chemical, those show up within Kibi. So this is like one of these things where you gain wisdom by doing more curation, is that Kibi has a lot of these instances of using pluralization to denote a class versus a single singular to denote a prototypical instance of that class. So there's a couple other kinds of like QC things that help identify places where there might be incorrect curation or like missing curation, and that gets auto-generated every time someone makes a commit to biomappings on the GitHub repository. And then they get turned into JSON files or YAML files, which get like automatically turned into a website based on a static site generator. So all these things are very transparent, and like anybody can grab those resources or look into them and say, hey, this is, maybe something I want to make 5 contributions today, and these would be valuable contributions to make. For the audience, the most important thing is we do all this mapping stuff. Let's use SESM. Let's like use the standard that we spend a lot of time building and evangelizing. So let's use that to have all of this really well-defined semantics, which means that all the prefixes are well-defined, all of the predicates, besides this example you guys maybe pointed out with the species-specific thing, all the predicates are well-defined using these standard predicates. The labels are included for all of the objects and the subjects and the mappings. The curator provenance is kept, like how it was generated. So if it was generated by a lexical matching, it keeps that information. Yeah, and so, there's pearls that go with this data that point to like the most recent version of it, either in TSV, JSON, or OWL, and then like this metadata file. So if you want to consume biomappings and put it into your workflow, you could just like suck it in with any standard workflow that uses SESM. And so this is supported in ODK now. And Nico could say more about this if he wants to later. There's like the Mapping Commons sort of framework for pulling in various mappings in different places. So you basically get this stuff for free. So yeah, the last bit of this is that, you know, Biomappings is sort of a new model for community curation, because I mentioned it's got this open code, open data, open infrastructure thing. You know, all the code is licensed really permissively. All of the curations are CC0, though they keep all the provenance information associated with them, so we can track people but based on their ORCID, and then like dole out co-authorships, you know, generously to people who curate content when we make papers about this. Yeah, and then we've got all the quality control stuff I mentioned before. And this this also, you know, puts the data onto Zenodo. It gives it a DOI. It builds a package that has the data inside it, so you can like import it as a Python package, you know, and all this kind of stuff. And so, yeah, anybody can basically go and curate something in Biomappings. And I'm going to show you now. We do the interactive demo. You can curate something, make a pull request, and then it becomes part of the Biomappings repository, and then everyone benefits from it. So yeah, I want to point out, yeah, Krishna, who is working on AgriVoc and agro ontologies, like, you know, he decided I want to use this workflow, and he did like, you know, 100, 150 curations between these two vocabularies, and this was awesome. So, okay, last thing, if you're like doing some super secret stuff, this probably isn't the right audience, but it's possible to also use Biomappings on custom files, so you don't have to be like locked into this community version of this. So this is maybe more interesting for people who are doing you know, projects that are behind like, you know, some kind of export controls, or maybe companies that want to do it themselves. So this is there in case you're interested in this. Sort of, I glossed over this, but by default, Biomappings is using the bioregistry as a standard for all of its prefixes and quality assurance. So this is good. It's like internally consistent. It's got a lot of really good tooling around it. It conflicts with some other standards that people think. So sometimes the capitalization is a little different, but that's okay. That's easy to overcome. Okay, so that's the end of the presentation. And so if you use this stuff, by the way, we have a publication, you can cite that, but you know, it's better to just look at the documentation than read a publication. Yeah, and the issue tracker is also right here. So I think by now you probably found your way to the repository. And yeah, so this is funded by like a DARPA program. So I'm going to stop there for a second, and I'm going to stop sharing. And then we're going to do this as an interactive demo. So let me grab the interactive demo stuff and copy paste it into the chat, just in case somebody hasn't got the slides themselves. And then we're going to like do all of it together. So the first thing I want everyone to think about is there a specific thing you want to curate right now? Like, does anyone have a favorite namespace? We could do this Mondo Mesh thing if you want, or we could do whatever comes up first and feels right. All right, so here we go. We're kind of back in my screen. We're going to go into the shell. I'm going to skip the first two steps. So the first step of this is to do, you know, git clone. Okay, so we want to git clone this. Actually, before we even do this git clone, you need to make a fork of this repository, because if you git clone the origin, you're not actually going to have rights to push onto it. So you're actually going to want to make your own fork, and then it's going to be something with your username in it. Maybe like for me, that'd be CT Hoyt. Question for the audience: can everybody like read my terminal, or is it kind of a problem because it's got some transparency?
No, we can read it fine.
Okay,
I can read it fine at least. Yeah.
Step one is to clone it. So you can follow along. This might be a little hard to follow along, because if we wait for everyone, it will take too long. After you clone it, you can CD into your Biomappings folder. Once you're here, let's just take a look. So we've got like all of our setup configuration for this Python package. We've got the source folders where all the code is. You know, the rest of this is extra configuration. The most important thing is we can do pip install -c. That stands for editable mode, or you can write it out in full, editable, and dot. So that means install the code that's in this this folder that we're in right now. And the last thing you need before you finish is to add square bracket web. And what this means is there's some optional dependencies that are necessary to run the web app. So you're going to want to install the optional dependencies, which includes Flask and the Bootstrap extension to Flask and a couple other things. So once you do that, you will be able to run Biomappings web, and -- help will give you some information. So this is on the last slide. You don't need any configuration. You just kind of run it, and then great stuff starts happening. So let's try that. And then we'll see what happens. So it is going to say it's running on localhost 5000. Depending on if you're on Windows or Mac, it might say like HTTP 127.0.0.1, but this is the same thing as localhost, so you can kind of use whichever one's more comfortable. All right, so here we go. We're in the curation interface. You know, it's sort of up to us to do whatever we want at this point, but the curation interface has the source, has the query and the name for the source, and it has the query and the name for the target. All of the mappings in here are assumed to be exact matches. So typically we want to give thumbs up to ones that are exactly the same. And if there's some question about that, if they're not exactly the same, or they're just completely different, we're going to give it a thumbs down. If we're not sure, we give it the question mark. That's what these all have on it. So also attached to this is a confidence. The confidence is sort of a relative thing. Depending on the script that generated the predictions, this confidence means something different. But you know, the rule of thumb is bigger is better. So yeah, it's kind of up to us to start curating things. So let's look for some examples and see how we can do. So for guanidino benzoic acid and for guanidino benzoate, you know, this is a tricky thing. Actually, we probably shouldn't start with chemistry curation. This is pretty tricky because the benzoic acid is like the form that has a hydrogen attached to it, and then a benzoate would be like the ions. So it really depends on who you are and what you think about chemistry mappings. So now that I'm seeing this, maybe we shouldn't be doing chemistry as our first demo. So why don't we go into the summarize button, which gives a high-level overview of all the predictions that we can curate. So maybe we want to spend some time curating, yeah, maybe mappings between mesh and human phenotype ontology, or mesh and EFO. And anyone have any preferences?
HPO.
HPO. You want to do mesh and HPO?
Sure.
Great. Yeah. All right. So here we go. So I clicked on that, and so as we're doing things in this web app, we're sort of accumulating things inside our address bar, like various filters. So this should, if you see it, it should look pretty obvious. Like source prefix is mesh and target prefix is HP. I don't have time to go through all the things that are there, and the web interface is very, very simple, so it doesn't have explanations of all of them. So there's a few hidden gems, which maybe you'll find yourself. So none of these look like really obviously the same. So it might be the case that we actually want to follow up on them. So because we have these standard queries, we can use the bioregistry to resolve them and go and look at the pages that are attached to them. So I don't know, I picked this one, ANA2 antibody. So the note says it's an antibody associated with paraneoplastic opsocolonis in a patient with large cell carcinoma of the lung. So okay, that's a mouthful. But you know, if we kind of put their stuff side by side, we might find out that they're the same thing. So this is the presence of autoantibodies in the blood circulation that react against RI, an anti-neuronal antibody. What do we think, team? Are these things the same, or are they not the same?
No, I think that this is also a very good example for where it's very, very difficult for someone without, you know, understanding the conceptual model of like HPO and
all right, here we go. So here's how I curate this. I realize these are not obvious enough to be the same, and I am not a domain expert. So I'm going to click, I'm not sure if this is correct or not. The good thing about this, I'm not sure if it's correct or not, is somebody later can go and look at this list of things that have been marked as unsure, because people don't use it so often. And it means that someone has looked at this. It's not like it's just been sitting there. So yeah, that's one possibility. All right, let's try another one, right? We want to see some success. Let's go to the next page. Anything standing out to anybody? All right, here's another actually a good example of when we're doing mappings. We've got achromatopsia 1 and achromatopsia. This is like a classic scenario where it's very difficult to know if these are a mapping or not, because this one makes me think maybe there's multiple achromatopsias, and maybe the relationship between these two things is like a hierarchical one and not necessarily an exact mapping. So I'm gonna open both of them, and we're gonna do the same thing. We're gonna look at them left and right. This is the wrong one, sorry. Mesh, yeah, Zoom is always putting its bar in the place where I want to click things on my web browser. Okay, so Yeah, unfortunately, it looks like the mesh record doesn't have a lot of stuff to say about this one. So it's in color vision defects. It could potentially be the same. But we probably want to look at the hierarchy. Okay, so this is in the mesh supplement. So because it's in the mesh supplement, there's no hierarchy associated with it. So here's like another example of something that's very difficult to curate. And you might be asking, like, curation's so hard. Why is this happening? And the reason is because we already did like several hundred curations of mesh and HP mappings. We did all the easy ones already. Like, okay, so let's skip this one, which is hard because I don't know the answer. Let's do one that looks pretty easy because it looks like it's the same. So partial.
Charlie, there's a couple questions on the chat if you want to maybe take a look at those real quick before you start another example.
Yeah, it's much. You guys feel free to please speak up because I can't look at the chat while I'm also doing this.
Yeah, sorry about that, Charlie.
Okay, so Harris, yeah, go ahead, Harry. You can ask.
Yeah, this is definitely a related question to the concept because I was just curious if there's a way to specify that kind of level of detail in the curation interface where you're saying, maybe it's not an exact match, but you know, I know of some other SCOS predicate that may actually be more appropriate here.
So the curation interface is really stupid simple. Like most people that build websites, they talk about using like JavaScript front ends. Like this has just got a back end that every time you click something, it links to a new page and it like reloads the whole website. So I haven't put that kind of interactivity. And so what might be like solving Harry's question is rather than just having a thumbs up and thumbs down, there could be like more buttons that like, these are a close match, or this is a, narrow or broader. So there could be like extra things on the dashboard here because there's sort of the implicit assumption that we want to look for exact matches. So the answer to Harry's question is there is no way to do that. But if that's something that people really find is useful, then it could be implemented relatively easily. Okay, and then Trish asked, if a mapping is marked as correct or incorrect by one user curator, can another user still curate that mapping and either mark it as correct or incorrect? That's an excellent example. So that's the next thing we should look at. So in the background, every time we click the thumbs up or the thumbs down, the Biomappings web interface and by sort of transitivity, the app that's running in the back is actually interacting with the Git repository of Biomappings. That's why we had to, up here, install an editable mode. So let me hop over to my GitHub desktop interface because I have respect for myself and don't use Git from the command line. So as we make curations, it has this file called predictions.tsv where all of the predicted mappings are. And it actually like takes the lines out of this predictions file and it puts them into one of the other files. So in this example with this ANA2 antibody, which we weren't sure about, it took it out of the predictions file and it put it into the unsure file. And when it did that, it added provenance information that it was me that did it, and it keeps the appropriate semap v, curation type to go with it. It also is sort of like a little bit extended. So these files aren't in the SESM format because it actually extends to more information than actually fits inside SESM. So that's fine. So it keeps track of the way that it was predicted as well. So let's come in here and let's just make a correct curation, and then maybe let's make it incorrect. So I think that the case for these, yeah, okay, well, let's just go back. So now you can see that these have changed a little bit. And so the predictions file now has 2 lines that have been deleted. And then the positive mappings file has the curation that I just did. And so the question was, if it's marked incorrect by one user, can another still curate that mapping and mark it as correct or incorrect? So the answer to that is no, not directly. The Biomappings interface doesn't expect people to curate things a second time. But these things live inside, these TSV files. And so it's totally under the purview of somebody to say, I want to open up this file and I want to find a line that I don't think is correct and I want to put it somewhere else. So this is maybe a philosophical decision for Biomappings to keep it simple, that like one curator is enough. There's definitely reasons why that might not be true. Maybe you have disagreements between curators. So there's no way around making some kind of assessment of the quality of the content you're pulling in. Yeah, I don't know. So we could extend the Biomappings workflow to have like multiple curators for the same statement. And so that would need like a different kind of data model. It will also like need a huge buy-in for people who want to recurate content instead of curate novel content. Because I mentioned before, there's like 40,000 predictions that we could be curating. And I don't know, it's hard to tell what's the benefit of recurating something that already exists versus curating something new. My idea is the drive-by curation. So one day someone's going to consume biomappings and they're going to find, hey, some analysis I did is wrong because there's a wrong mapping in biomappings. And when that happens, I want someone to come and make a drive-by curation where they fix one line. And that's going to be the solution to this. That's the happy medium, in my opinion.
But a drive-by curation will require this to be a web app with some kind of authentication associated with it, not just something you will run locally, right?
I mean, you can like go onto GitHub, and then you can go into biopragmatics, biomappings, and then you can like open up this directory that all the resources are in, and like these TSV files, they're versioned on GitHub. Like this is the single source of truth is the GitHub thing. So it's kind of like up to you. You can open up one of these files, and actually they might be too big to open on GitHub, and you can just, you can edit the file. You can say, hey, that line's wrong, so let's delete it. So that's the happy medium version of this. Like for sure, there's immediately the desire to say, hey, why is this web interface? Like, why do I have to run it locally? Why isn't there some hosted service somewhere that takes care of versioning all this? And like, yeah, I get it. That would be great. But this is like something that was easy to create. And for me as a curator, I use this all the time. I can do, hundreds of curations per hour in topic areas where I'm like knowledgeable, not necessarily in topic areas where I'm not knowledgeable. And this is already way faster than other things. So yeah, there's always the question of whether we could have a more like generic solution. Every time James Overton gives one of his talks on some of his tools for doing like web-based table editing that also maps into Git, I think that's like, that could be a way to do this. Maybe people have ideas about this. I don't know. I wonder like to what extent there'd be a lot of users that are interested in this that are unable to like git clone and then pip install stuff. I mean, I know that this is a barrier for some people, but in the ontology world, like those people are typically working around at least one person who knows how to do this stuff. So I'm curious if you're in that situation where you think like the code that I've sent in this like five lines of git cloning and this kind of thing, if that's a barrier for you, like we should talk about that.
Yeah, I think to actually make this scale to like a wider range of people to actually do drive-by curation, you would have to integrate some system like KGCL inside of OLS and these kinds of browsers, so that people can just say, okay, this mapping that I see here in OLS is bad. I just click a button and it will immediately just make a pull request at the source of the mapping set that is listed as a source for that mapping. And I think, like, I guess that this is my next, that would be the next way to next level to scale this up, is make that super, super easy and not having them to use actually Git workflows. I mean, I don't know, like maybe it's possible that 20% of us will do a drive-by curation like this, but all of us, I would say we need to integrate.
Hold on. I wouldn't say that doing like curation with this interface is drive-by curation. I would say that's sort of a more substantial curation. But if you say like, oh, there's something in biomappings that's wrong, like going and marking it as wrong and deleting it from a file, I think that's like a drive-by curation. Okay. But I get what you're saying. There's definitely technical barriers to like doing this without having some more complicated system. But that does bring me to the next part of the workflow, which is how you can take biomappings and contribute it back. Does anyone have any other questions about this this web interface right now? I mean, like it's pretty simple. You can also have it interact with Git on your behalf. Like you can do this commit and push button. I've got some stuff that's in my working directory that's not related to this, and I've also only been working on the master branch. So typically you want to open up a new branch first when you're making some contributions, and then you can you can follow up on that branch instead of the main branch. So we could actually do that right now. So okay. So question for the audience again, what would be more interesting? Do you want to see how you can generate more mappings yourself, or do you want to go directly to how to contribute them back into one of the ontologies?
I would like both, but maybe start with generate a little bit just to, so I guess for the lexical side, so is there any, do you use any kind of more, so you've described your lexical approach a little bit before, but Is it something that's also usable in isolation as a tool? Is this all Gilda? What exactly is the tool that is doing your lexical predictions?
Sure. Okay, it's time. We're now going to go into a little bit of code walking. So hopefully the stuff that I'm showing you, it's like very high level. And then if you want to use this to do more powerful things, you can go into the guts of it. So let me bring up my PyCharm and get out of SessimPy, my current like massive project of refactoring. So this is my project. Okay, so let's see, we're in the wrong one. Biomappings. So I've got this opened up, and there's a scripts folder, and inside the scripts folder there are many different scripts for generating various mappings. And maybe before I show you one of those, I want to open up the predictions file and just show you some lines from the predictions file. So every prediction has a reference to the file that generated the prediction. So in theory, I'm not going to accept any contributions to biomappings that create predictions. That doesn't also include the script that generates the predictions. I really don't want to have this content that we don't know where it came from or how it came from it. The good news is to generate predictions with these scripts, it's like relatively easy. So let's go into this list of all the scripts. Why don't I open up Uberon to BTO? Okay, so here's our script that generates the mappings from Uberon to BTO. This is in theory like a two-line script. I don't know, it really depends how you count it, but there's really one line that does all the important stuff. There's this high-level workflow called append Gilda predictions. And so you give it the resource that you want to do kind of the mappings around, and then the resources you want to map it to. So maybe today we want to map like not just Uberon to BTO, but maybe we want to map it to mesh as well. So this would be it. And then this provenance thing like gets the script, the URL for the script. So this is a little bit meta, but you just have to kind of reuse this by looking at, one of the many. Oh, that one's a little complicated. Yeah. So this one's a little bit different, but a lot of these mapping workflows are basically the same. So yeah, when you run this, this function append Gilda predictions, it wraps Gilda and it does all this lexical matching based on the resource. It also uses PyObo to get the content. So it uses PyObo to get Uberon and BTO and mesh. PyObo is like this very generic library that uses a combination of readers for obo files and owl files, and also like custom implemented sources to get other databases as ontologies like mesh. Yeah, and so this is sort of generic, so you just need to know the prefix that goes with all the things, and then it looks up that information about how to find those resources using bioregistry, and then it does all the stuff in the background. So that's the highest level version of this, and then you can kind of go and let's look at like an example of one that Ben wrote that really directly it gets an obo file and then it does its own specific logic on that. So it's kind of like up to you to build up the mappings. In the end, Biomappings gives this data structure called the prediction tuple, and you just have to fill up the prediction tuple. So this corresponds to one row in the predictions file. So those are the two like ends of the spectrum, is like the canned pipeline and then the totally custom pipeline. So in case you want to know a little bit about how the canned pipeline works, the append Gilda predictions one, its job is to get a grounder from the given target prefixes. So get grounder, this is also part of Gilda. Hold on, so the get grounder function is, see, where does that live? I think that's actually inside this file. So now we're inside Biomappings right now, so it has some utilities for working with Gilda. And the get grounder function, okay, this actually lives in PyObo. So the job of that is to use PyObo's system for generically looking up ontology-like resources and then building up a Gilda index. So I don't know, I kind of want to stop there because it's getting a little bit into the weeds. But the point is, there's this high-level functionality implemented in Biomappings that uses some combination of PyObo and robot for getting ontology resources and other resources, and then it loads that up into Gilda for you. So you can sort of follow that code to figure out if you want to build your own Gilda indices, like in other ways.
But in theory, these you could basically use any approach you would like for generating the prediction. So you're kind of, the framework here just says, okay, every kind of resource to resource prediction has one of these Py files, and then
you can do whatever you want here. So if you want to call oak and you want to use Oak's lexical
exactly.
mapping, you can do that. Or if you want to call PyKeen, I think I don't have the example in here because the PyKeen like machine learning based pipeline is still kind of under development, but like using that to develop to create the predictions is also another possibility. So it's up to you. You can make them however you want.
And and from your experience, the fact that every kind of combination of resources gets its own Python file is basically based on the fact that very often you do want to build in some kind of like knowledge about, let's say, the lexical weird regularities that a resource has, and just use that to pre-process labels and stuff like that, or why?
It's really up to you. There's definitely scenarios where we've done multiple kinds of mappings together. Each of these files typically corresponds to like one day I said I need mappings for this, and so I put it there. There's examples where we generated many mappings together at the same time. So maybe we try and find an example of that. Yeah. So example for DOID. When we were doing this, we wanted UML as EFO and mesh, and then we had some other stuff going on that was making it difficult to do other mappings. So it's really up to you. If people want to make one file for every like pair of things, that's fine. But if you want to say, I want to generate all the mappings for Mondo to everything and have that be in the single file, then that's also fine.
Okay. But the point is, we have a Python file. This is documentation. So it's provenance documentation that you can always point to and say, okay, this is the source of my approach of how I generated the predictions. Yeah. Okay, good.
And that gets stored inside the predictions. By the way, if people want to make predictions with things other than Python files, I'm totally happy with that. Like, you just have to make rows in a TSV file. So if you can manage to add rows to a TSV file correctly, like with your not Python file, then that's fine. So yeah, this is totally generic. All right. Let's see what else is going on in the chat. Okay, so that was generating the mappings. I want to now tell you about like this. This isn't a very well thought out workflow, but we have a couple scripts that have gone through and we've done something really ugly. So here's a script. This is the V1 of the script that goes through the Mondo file, and it's an obo flat file. That's the Mondo edit file, and it goes through and it kind of like figures out the place where to put the mappings inside there using some like string logic. And I hope everybody's really cringing at this because it makes me cringe too. Ideally, the way that we deal with this would be to use KGCL and to generate KGCL and then just to apply that directly onto your files, especially because working with OWL functional and working with like OWL RDF is like near impossible. It's really, really pain in the butt. So if you must do that, the last thing I want to show you inside our thing is I've generalized the script for making contributions to obo files. I almost have got it to work for OFN, but I don't know, maybe this will never work. And it's probably not worth like my time to finish it. So there is a script that lets you like contribute back into the Mondo edit file or any other obo flat file. So I'm not going to do that now, but like, that exists. If you want to try it, you can like get in touch with me. So we've used that to make contributions to like several resources and different flavors of this. So yeah, people are pretty happy where it's like, hey, we managed to curate like 500 mappings from your resource to like external resources, because once you have this curation interface, it actually goes pretty fast for the easy ones. The hard ones take a little bit longer, as we all found together by doing this. Okay, so I think that's it. I think I'm at the end kind of the demo. So you all saw how to install biomappings and run it yourself, how to filter down to like some of the mappings that you want. I think some of the other parts, I didn't mention like the search bar, but I think you'll be able to figure out how to use the search bar yourself when you run it. If you want to make mappings, you can always like get in touch with me and we can make them together. We can generate mappings for resources that aren't available through PyObo already. Typically, the way that we do that is add new resources to PyObo. So like this canned script can work. Yeah, so if anybody's got like some sort of general questions, we got 4 minutes left before the top of the hour.
Before we start with the question, if anyone has any remaining ones, I think many of them you have already answered. I just want to say really thank you. This was very, very illuminating also for our kind of, collaboration on all these kind of different tools that should all work nicely together. So it's very good to see all this stuff. And yeah, just heartfelt thank you. And if anyone else wants to ask any last questions, general questions, visionary questions, or simple questions, just go ahead. We have a few more minutes left.
Yeah, you're welcome. If no one has any questions, then I have one other slide to show you. But please start with questions, and I'll only give the slide if there's no questions, because the slide's going to create a lot more questions than it answers.
Just put it. We have only 60
Yeah,
seconds left.
I said it. I have to put it now. Okay, so what's next, right? I told you I wasn't going to talk about large scale assembly of mappings, but like in the end, we don't just want the SESM file from Biomappings. We want the SESM files from every ontology, and we want it from Wikidata, and we want it from, you know, UMLS, even though they're evil bad people at UMLS who keep their stuff hidden behind bad licenses. Sorry, they're not evil. They're just negligent or something. Don't quote me. It's recorded. Anyway, it's on my blog post. I already said all these bad things. But anyway, we want to be able to get all this stuff together. We want to be able to reason over it. So like align, deduplicate, assess the confidence, filter out bad stuff. And then we want to turn this into stuff that we can reuse directly in our pipelines for building knowledge graphs, for building named entity recognition systems, for building up databases, for just exploring mappings and understanding how they're created. So SEMRA is my new project where I'm working on building that right now. And it's kind of all working. It's just not up to the standard of my normal code, which has documentation and tutorials and testing with everything. So yeah, that's how it starts sometimes. So how does this like look in practice? The cell line thing I showed you, you've got like EFO, Cellosaurus, DepMap, CCLE, and biomappings. And you want to be able to like align all those together. You want to get rid of stuff that's irrelevant. So maybe there's mappings in EFO going to like OB. We don't care about that today. So we're going to filter out those mappings. We want to make sure we get rid of anything we know is a negative mapping. Anything that's like, self-match. And then sometimes we know like, yeah, in EFO, all of the mappings to mesh, they're actually exact matches, even though they're not written as such. And then we're going to do inverse, and we're going to infer inverses. We're going to do all sorts of other reasoning, like inferring chains. So like 2 exact match is a chain of that creates another exact match, a chain of exact match to broad map, that's a broad match that creates a broad match. Yeah, and then we're going to do some more filtering, and we're going to consolidate and export and do all this stuff. So this is kind of like where we're going with this, because once you build this sort of externally consistent mapping database, you can use it in your use case. So anyway,
Very exciting, Charlie. Very good.
That's
Very
what I'm
good.
thinking about right now.
Very good, very good. We will definitely talk about that a bit more in the future. So, and everyone else that's interested, but now we have to close. Thank you very, very much, and talk to you soon.
Thanks, everyone.
Thank you.